neurodegenerative diseases.
Thank you. Good evening, everyone. Thanks for the opportunity. So, what we do at the bioinformatics group at Scuola Normale Superiore in the BioSNS laboratory, we are passionate about understanding biological mechanisms by integrating information from sequence, structure, and protein interaction network to uncover new mechanism of function of biological systems. And we use in turn this mechanistic knowledge to interpret large-scale omics data set, in particular genome sequencing and transcriptomic data set, oftentimes in personalized medicine settings. And we are also interested in using AI and data-driven approach to discover new precision therapies as well as engineering chemogenetic tools. We are particularly interested in systems involved in signal transduction, in particular G-protein kappa receptor, with a special focus in cancer. GPCR are the largest family of transmembrane protein, which account for roughly 1/3 of the approved drugs in the market, so they are hugely important from a pharmaceutical point of view. And from a biological point of view, they transduce a multitude of extracellular signals inside the cell by coupling to intracellular heterotrimeric G protein, okay, that are grouped into 4 different families, accounting for regulation of different intracellular pathways. Understanding of these mechanisms is extremely important because approximately 1/3 of G-protein kappa receptor lack this mechanistic information. So in recent years, we developed machine learning and artificial intelligence based pipeline to uncover the sequence and structural determinant of G-protein kappa receptor at a human proteome-wide scale. And this information is extremely useful to better understand the signaling properties of this receptor, which might also help the interpretation of omics dataset, transcriptomic dataset, and ultimately can also guide the design of new chemogenetic receptors. However, we are increasingly interested also in exploring and modeling the network, the big network that regulate extracellular signaling of G-protein kappa receptors. Why? Because it's emerging as a major player in mediating the interaction between the cancer cell and the tumor microenvironment. And in this context, it's it's wiring a very complex cell-cell communication network. And the overarching goal in this case is to leverage the huge arsenal of approved drugs available for this target for repurposing in cancer. To this end, we have recently compiled a very large network of GPCR and their cognate ligands, as well biosynthetic enzymes that synthesize organic ligand. This information is extremely important because we plan to use biosynthetic enzymes as a sort of proxy to estimate the activity of organic ligands that represent approximately half of the ligand of GPCRs, and they are not genetically encoded, so they are not present in genome sequencing and transcriptomic dataset. And this information could be extremely useful as a complementation for the analysis of cell-cell communication in single-cell RNA-seq or spatial transcriptomics dataset. We use this information to mine publicly available large-scale cancer transcriptomic dataset And we found out that many of these GPCR axes entailing either the conjugate ligand or biosynthetic enzymes are significantly associated to patients' survival and are concordantly differentially expressed, meaning that the concordant differential expression of these axes is more strongly associated to patient survival than the individual component of the axis. And we compiled a long list of these significant instances across cancer type, okay, both for receptor ligand and receptor enzymes. In our analysis, we also integrated information from pharmacogenomic dataset. Okay, so these datasets, such as the genomics of drug sensitivity in cancer, or GDSC, and PRISM, essentially consist on profiling hundreds of cancer cell lines for 100 in the case of GDSC, or thousands in the case of PRISM, oncological and non-oncological drugs, respectively. Okay, so it is a huge repertoire of drug sensitivity information suggesting which drugs lead to higher sensitivity of cancer cell lines, both for oncological and non-oncological drugs. We were particularly interested in PRISM because it contains information about many approved non-oncological GPCR drugs, and we found out that GPCR drugs that significantly inhibit the cancer cell growth indeed target the axis, the prognostic axis that we described before, here indicated by this red dot on the right top corner of this scatter plot. We also validated some of these compounds, in particular the antagonist of adenosine receptor in hepatoma cell lines, and we are extending this validation also to additional cancer cell lines in hepatocellular carcinoma and cholangiocarcinoma, as a part also of a collaboration with the group of Fabio Marra and Chiara Raggi here in Firenze. But what I want to stress is the emerging, that this data also supported the emerging role of neurotransmitters and conjugate receptor for neurotransmitters in supporting the oncogenic growth of cancer, since many of these drugs are targeting receptors for neurotransmitter. We use the information, the big data available in the PRISM and GDSC dataset also to set up machine learning models of drug sensitivity. And this work was done by a highly talented student in the group, Francesco Carli. So essentially, we trained drug-specific models, so totaling more than 6,000 drug models, combining GDSC and PRISM, that were able to predict the drug response in terms of cancer cell line growth inhibition as a function of cancer cell line transcriptomic data provided from RNA-seq of the cell lines. And this was achieved by a massive effort, training effort carried out at the Leonardo Cineca Supercomputing Center. The models that we obtain are comparable to the state of the art in this field, but what I want to stress here is that they are interpretable, okay? So we used an algorithm that is inherently interpretable, and by mining the gene deemed more important by the model for the drug sensitivity prediction, we could actually demonstrate that the model is learning drug mechanism of action. Okay, so it's not learning from random noise, but is learning by itself, starting from an unbiased, from the whole genome profiling, whole transcriptome profiling of the cell lines. Those gene whose expression is more important to mediate the effect of the drugs. Today, to cut a long story short, I wanted to focus on the more translational aspect of our recent effort. So you might wonder, how can we actually exploit a model that has been trained on cancer cell line on real patient data set for predicting drug repurposing strategies. So our take on this is to use a cell liner. Cell liner is an algorithm developed by the Broad Institute that transform cancer cell line RNA-seq data and bulk RNA-seq data from patients in a common latent space that allows the alignment of the two types of transcriptomic data. Okay. After this procedure, we have run the inference with our cell-lit model for the entire transcriptomic dataset from TCGA, the Cancer Genome Atlas, as well as for a specific cohort, patient's cohort, one of pancreatic cancer provided by Gioacchino Natoli from Yale, and one for glioblastoma multiforme provided by the group of Chiara Mazzanti from Fondazione Pisana delle Scienze. What you can do with inference with our model? First of all, you can rank your patients that are predicted to be more sensitive toward a certain cancer drug or non-cancer drug. Also, you can take the predicted sensitivity, so you can get for each patient a sort of sensitivity or responsiveness profile predicted for all the drugs for which we have developed a model, and then use this profile to cluster, to compare the patients, okay? So first of all, we have demonstrated that our model, cell-lit, is able to recapitulate cancer drug prescription. So we ran the inference for all TCGA samples, and then we considered the top-ranked samples for each drug model, okay, showing that 90% of the GDSC drug model recovered among the top-ranked sample at least one patient affected by cancer type matching the drug's prescription, okay? And this is indicated by this stacked bar plot here. Of course, certain drugs recover pretty well the matched cancer type for that specific drug. And this represented a sort of validation of our approach. However, our modeling generated a huge number of additional hypotheses that potentially are all potential drug repurposing hit, okay? Both for monotherapy, including again GDSC drugs, so each drug model generated prediction for many other cancer types for which the drug is not yet prescribed, here indicated by stack without asterisk. Of course, we have tons of data of prediction for PRISM, non-oncological drug. This is the bulk of our drug repurposing effort. And we were also able to recover drug combination prediction, okay? Since there were many instances where we predicted for the same pool of patients multiple drugs, okay? Here indicated, so the red circle indicate approved, known approved drug prescription, while instead the other instances we have some evidence in the literature, but these are not yet been approved. And the number of these dots represented the number of samples for which we had this type of prediction, okay? The last example, we used the cell-lit profiling for cohort of patients provided by collaborator. Here I'm just focusing on glioblastoma multiforme, but we had also pretty interesting data for pancreatic cancer. As I anticipated to you, we use our model to do a patient-wise profiling of each drug. Okay, so in this heat map, each row is a patient sample, and each column is a drug. Okay, and with this numerical profiling that you get from the model, you can cluster the patients. So you can see from this dendrogram on the left that there are actually two groups that are identified by our approach, that are characterized by slightly different overall sensitivity profile. Okay, so The upper cluster, the larger cluster, cluster 1, is characterized by slightly higher blue in this region than cluster 2 that is a bit more reddish. Okay? So we first validated our prediction, and we took representative sample for cluster 1 and cluster 2, and experiment on primary cell line actually confirmed our prediction. But what was more interesting was that by integrating the information of oncodriver mutation from whole exome sequencing on these samples, so that the heat map is the same as before, we just added oncodriver mutation here on the left, okay? And we could actually see that the larger cluster, cluster 1, was characterized by the higher burden of mutation compared to cluster 2, okay? Roughly half. Cluster 2 is characterized by roughly half of the mutational burden than cluster 1. So predicted higher responsiveness might be associated also to higher vulnerability of those samples with higher rate of mutation. So these are the conclusions. So I hope to have convinced you that GPCRs could be promising target for oncology. And a story that I haven't told you is that we are exploring also their role for immunotargeting, okay, which is an emerging field of GPCR role potential in cancer. And we have developed this model, CELIT, that provides inference. Let's infer the most sensitive most effective drug for a given sample, and we plan to release a web server for this model soon, by the end of the year. And we have generated a lot of drug repurposing hits for mono and drug combination. And this is it, so I conclude, and I want to thank all my group and collaborators, and also the sponsors for their generous support, including the Tuscany Health Ecosystem. Thank you for the attention.
Thank you. Any questions from the audience? No, I have just one remark for you. I mean, your model seems to be very powerful, and actually it seems like a business model could be associated with that, related to the fact that, for instance, you predict some combinations of new drugs and repurposing. Have you started reasoning on this side of the story to say, to build a business model out of this, because it's kind of valorizing your results?
See, you mean for the combination?
Yes.
Yeah.
The predictive of the combination and.
So.
And the
repurposing. Combination are emerging naturally from the inference. So we haven't trained the model for the task of predicting drug combination. Although this profiling, this cancer cell line profiling, are now also including for certain tissue, for cell lines of certain tissue, also the drug combination. From our inference, we simply realized that there were many instances where pool of samples had multiple instances of drug predicted that were more sensitive for multiple drug predicted drugs. So there seemed to be a high potential for that, and possibly this can be further reinforced by training for this specific task of drug combination. Or also another very cool, I hadn't had the time to mention this, another very cool line of research is combination with immunotherapy. That so targeting, also GPCR, I mean, there are now solid evidences that targeting certain GPCR axes might indeed restore and sustain the response to immunotherapy. So yeah, the combination offers a lot of opportunities, drug combination.
Byproduct now.
Yeah, but maybe very promising byproduct.
Thank you.
Thank you.